Welcome to another episode of the BioHub. Today, I'm joined by Andy Parker, the CEO of Step Pharma. Andy has spent over 2 decades in Biotech and Pharma, holding senior leadership roles at companies like AstraZeneca, Shire and Zealand Pharma, and even spending time on the VC time at Eclosion. Since 2019, he's been leading Step Pharma, a company developing first-in-class CTPS1 inhibitors, with their lead program now in the clinic for cancer. We talk about the science behind Step's approach, what's next for the company, and Andy's experience moving from big pharma to small biotech, plus lessons on fundraising in today's market and building a great work culture. It's an absolute pleasure to have you, Andy. How are you?
Very good. Thanks, Max, for inviting me.
No, it's amazing. So you've had a pretty interesting path. You know, we talked about AZ, Shire, and also VC. What then led you on to Step Pharma?
Yeah, I think I've been very lucky to have worked across a broad spectrum of companies, you know, from that big pharma to the mid-size and startup biotech as well as VC. So that's been a privilege. I think I've also been very lucky to have worked across a number of different countries, which has exposed me to different ways to do things, trying to pick the best from each of those across the US, the UK, Denmark, Switzerland, etc. My preference, having been through those experiences, is clearly to be working in a small biotech environments. And I was really attracted to Step Pharma because of the underlying mechanism, so the human genetic story that validates CTPS1 as a target. Also because there were a lot of my former AstraZeneca colleagues at Signature in Nottingham who had run all of the medicinal chemistry in the early days of the company to get to that lead candidate. And a fantastic syndicate of investors who were very much behind the company. So all of those things were really attractive when presented with the opportunity to pick up the reins and lead the company from 2019 onwards, bearing in mind that the company was founded in 2015. So it had already been running for a few years and was at the point of having that clinical candidate identified by the time I came in. So I could really see a clear path forward to build a team, to take the program on through IND enabling studies and into the clinic. So joined at an exciting time with a really interesting mechanism.
That's awesome. And those for people who don't know who Step are, what's the core of what you're building?
So we're a French biotech company. We're headquartered in Paris, but physically located in Saint-Genis, which is very close to Geneva, Switzerland. And we are essentially a single mechanism company. So we focus on developing inhibitors of the enzyme CTP synthase 1. This is a first-in-class approach. So nobody else out there has a CTPS1 inhibitor close to the clinic. And in fact, there are not that many activities happening preclinically. So we're really sort of trailblazing this first-in-class opportunity. The reason it's interesting initially comes from human genetics. So the founding scientist, Alain Fischer at the Imagine Institute, identified a founder population of kids in the UK that had a profound inability to fight a viral infection. When they looked at the lymphocytes in those individuals, that immune cell repertoire was perfectly normal. But when they tried to activate those cells ex vivo to make them proliferate as if they were going to mount an immune response, they wouldn't proliferate. So that explained why these kids couldn't mount the response to the viral infection. And then using genetics, they established that the cause of that phenotype was a mutation in the gene that codes for CTP synthase 1. So it was essentially a null mutation that really taught us that if you lack CTP synthase 1, you have this profound inability for your lymphocytes to proliferate. But what was really interesting about the observation was that there was nothing else wrong in these kids. So every other cell type in the body, in the absence of CTPS1, could actually use an orthologous enzyme called CTP synthase 2 to make their CTP. And so that was really the genesis of the company. Could we develop a truly selective CTPS1 inhibitor that would block aberrant lymphocyte proliferation and enable you to then target that pathway very selectively from a lymphoma perspective? And that's really how we've gone on to sort of develop the company and enter the lymphoma space as our first clinical trial. But it's become much more interesting since then, because I would say what we've done is the genetics pointed us to this phenotype around lymphocytes, but we've followed the science all the way through as we entered the clinic in 2022 and began to ask ourselves questions about where else could you use this mechanism? How could you use this mechanism in solid tumors? So in solid tumors, we would argue that solid tumors express both CTPS1 and CTPS2. Therefore, if we inhibited CTPS1, they would use CTPS2 to make their CTP. So not a sensible place to play, as opposed to the lymphocyte population where we know CTPS2 can't complement for the loss of CTPS1. But we asked the question, are there solid tumors that have deleted CTPS2? And the answer to that question is yes, there are a whole range of solid tumor types that have deleted CTPS2, which creates this absolute dependency for the tumor on CTP synthase 1. And so we've moved now towards solid tumors, and we've started a clinical trial in solid tumors where we select patients for the loss of CTP synthase 2. And our plan right now is to focus on ovarian, endometrial, and lung cancer, where the prevalence of the loss of CTPS2 is between 15 and 20%. So there's a high prevalence of the loss of CTPS2 in tumors. We estimate 1 in 10 solid tumors have deleted CTPS2. And then we've also followed the science from the lymphoma study in understanding that as we dosed more and more patients through the escalation, we only have one recurrent adverse event, and that is thrombocytopenia. So we have an effect on platelets, and in order to mitigate that effect on platelets, we developed a schedule to dose the patients 1 week on drug, 1 week off, so that we mitigate that platelet drop. But as we built out the data set, we realized that we did not see any other recurrent adverse events. So we asked the question, could that safety signal be turned into a therapeutic endpoint? And the answer is, in the benign hematological condition, essential thrombocythemia, it can, because this is a condition where there's an overproduction of platelets. And what you need to do is to normalize platelet numbers in that patient population in order to help them avoid the issues that come from elevated platelets, which is thrombotic events. So now we're just about to start a study in essential thrombocythemia where we'll dose the drug very differently. But I think the important thing is we've really followed the science. Genetics took us into the lymphoproliferative space. Then understanding the loss of CTPS2 took us into solid tumor space. And then understanding our profile for our drug took us into the ET space. So the drug is called dencatistat. It's the first-in-class CTPS1 inhibitor, and it's well advanced now in its phase 1 escalation in lymphoma and its phase 1 escalation in solid tumors, and about to start in the essential thrombocythemia space. So we aim to maintain our position as world leaders in developing CTPS1 inhibitors, primarily focused on dencatistat as the lead asset.
Yeah, it's been a cool journey so far, like to get to the point you are now, and obviously getting to the clinic was like massive milestone. What's next then? What's next for Step, and then what milestones are you looking at next as big ones?
Yeah, so I would say for each of the indications, there are sort of big things coming over the next 12, 18 months. So in the lymphoma study, we're recruiting both T and B cell lymphoma patients. We just received orphan drug designation for T cell lymphoma. We'll be following that up with more regulatory interactions, most notably an end of phase 1 meeting with the FDA that will come sometime in the first half of next year. So moving forward then into phase 2, most likely focused on T cell lymphoma in phase 2, so peripheral and cutaneous T cell lymphoma. In the solid tumor space, dose escalation has been in all comers. So by the end of this year, we will be into CTPS2 null selected patients, both lung, endometrial, and ovarian. So throughout the course of next year, we'll be building the clinical data to demonstrate that we have clinical activity in those patients, and more importantly, to develop the concept of pan-cancer opportunity for this mechanism, because it works in ovarian cancers that are CTPS2 null, lung cancers that are CTPS2 null. It should work in any cancer where you can determine that they are CTPS2 null. So in the end, we'll be looking for that tumor agnostic approval. But throughout the course of next year, that data set will come and confirm the hypothesis and build out that pan-cancer opportunity. And then in essential thrombocythemia, we will start dosing patients in the next few weeks. And we will get a very quick proof of concept there, because what we're looking for is platelet numbers to be normalized. And so it's a relatively simple readout. We expect the effects to come on pretty quickly. So probably in the next 6 to 9 months, we'll start to understand whether we have a good drug for ET in our hands. So there's a lot coming over the next 12 to 18 months in terms of demonstrating proof of concept in multiple indications.
That's amazing. That's so cool. So when you look at like the big picture for Step, like what's the long-term goal? What do you want Step to become?
I think we have to ensure that we maintain our leadership targeting CTPS1. And then I think the most important thing is that we are efficient in running our clinical studies to, as quickly as possible, demonstrate the benefit to patients in hard to treat cancers. And maybe most importantly, build out the organization to enable us to do that, but also find in the long term the right partner that can actually realize the ambition here, which is to develop this drug across multiple cancer types and potentially as a pan-cancer therapeutic in the solid tumor space. And I'm conscious that that is likely to require us to have partners to do that, partners to help us with the development of the companion diagnostic that would run alongside this, partners to help us with financing the breadth of activities that would need to be undertaken to demonstrate the pan-cancer potential here. So I think that's the key. Let's grow the company to deliver on the phase 2s, and then let's be mindful of how we're going to really manage this to expand enormously in the phase 3 setting in the long run.
Yeah, nice. And are you looking at like those partnerships now for the future, and like evaluating? Is that how early you look at it?
I think we're always talking to people about opportunities. I think we are still at a point now where we need to firm up on the data set to really demonstrate that the hypotheses we have around the solid tumors and around ET hold true. And then I think the opportunity to partner will become much more straightforward as we move on.
Yeah. And then rather than the deal making side, like, everyone knows the industry is pretty tough at the moment, and fundraising is, I mean, pretty brutal. How have you approached it, and what's been key to you getting these fundraisers over the line?
Yeah, so we're in a Series C financing mode right now. We're hoping that we'll close that round out before the end of the year. We're making very good progress. I think the key to making progress is really the clarity of the science, the clarity of the messaging around the value inflection points that will be delivered through the financing that's being proposed. And then if you have good insider support, then that significantly eases the conversations that you're going to have with people around trying to raise money. And then persistence. I think that's also important. A no invariably doesn't really mean no. So the number of people that I have talked to where a no initially, when you continue to update them, continue to bring new data, etc., moves from a no to a maybe, and then with a very few, ultimately you convert those maybes into a yes. So I think you have to be persistent. It's a long game. In terms of fundraising, you have to talk to an awful lot of people, you have to build a lot of relationships, and you have to accept that no doesn't really always mean no. So I think that's key, because it takes a long time, particularly in this climate, to actually close a financial round. My only advice would be don't give up, don't get despondent around the no's, keep going back, keep the relationship strong, because those no's will convert over time, especially as data matures. I think it's probably much more difficult if you're a preclinical company, but as a clinical company for us, as the data has matured over the course of the last 6 and 12 months, those conversations have very definitely moved from no's to maybes, and in some cases to yes. And as I say, we hope that we'll close the round out and be finished with this financing effort by the end of the year.
Amazing. And do you think, you might put you on the spot here a little bit, but in terms of the market, do you think it's changing, getting better?
I think it's always difficult. I think it's been, if I reflect back on the last round, it was easier to raise money 4 years ago than it is now. It has taken me a lot longer. It has required an interaction with many more funds. So I think it is a tough environment right now. I think that, fads change, people move in terms of the things that they're looking for. Oncology particularly is now much more difficult because of the plethora of opportunities there are to invest in. So trying to find and demonstrate the differentiation you have compared to others, for instance, us being first in class, is definitely a positive. And trying to show that you de-risk is going to be key. So again, for us, having 3 shots on goal with 3 different indications is quite different from a binary opportunity, 1 molecule, 1 indication. So all of those things I think investors are looking for. But yeah, it's not easy, and I'm not sure it's going to get any easier quickly right now.
Yeah, unfortunately, that's kind of what I'm seeing as well. There's just so many uncertainties, right? Like in the market, and people just don't necessarily know which way to go. So one of the main phrases I keep hearing is data is key. Like you just said it then. It's easy being a clinical company. You can back up your results, and without being too brutal, but I suppose there was too many mistakes in that 2020, 2021 period where investors put big rounds in and they didn't come to fruition. So then they're more cautious, and then there's a political environment that's a bit unstable, and it's there's just a few factors that have just aligned, right?
Yeah, I think there were a lot of platform companies that people invested in based primarily on preclinical early data that didn't pan out. So that has burnt some people. I think in the oncology space and in other areas, small molecules seem to be not in trend. Although I think it's also important to remember that small molecules still are a massive component of approved drugs and a massive component of drugs in clinical development pipelines. But in oncology, you can see the fad towards ADCs and towards radioligands and away from small molecules. So I'm always keen to emphasize that there are many benefits to a small molecule as a drug versus going after an antibody as an ADC or an antibody or peptide as a radioligand. We're cheap to make, we're convenient to take. And if we get the molecule right, then you've got a very specific and selective approach to target any element of biology, in our case, tumors, but in other cases, in the autoimmune space, in CNS, etc., you still have that. So let's not forget small molecules, even though the trend right now is to go for in other directions.
Yeah, definitely. It's these trends are funny. I always think that a modality can go from hero to zero so quickly. For example, like cell therapy, everyone was on it, and there was so many big private raises in cell therapy, and no one wants to even touch it now. Even from a recruitment standpoint, every candidate wanted to be involved in cell therapy, even if they hadn't necessarily had the experience before, which you could get away with because it was a relatively new field. But now it's just gone the complete opposite way. It's crazy how it can literally go. And the VCs kind of follow these trends a lot.
Yeah, I mean, CAR T has been an area where there's been significant investment in oncology. It's certainly shown significant benefit in certain instances. So there are some diseases where CAR T is really having a huge impact. But there are others where it hasn't been as successful. I think people forget that there is quite a significant relapse rate following CAR T. And you've also still got to deal with the cytokine issues that go with treating patients. So whilst in certain situations it's a fantastic curative therapy, in many other situations it hasn't quite delivered that. And then the expense of treating people with cell therapies versus conventional therapies, from antibodies to small molecules. It's quite a different business model. I mean, the same is true of gene therapy, not an area I know very much about, but it's similar in terms of the huge expense to treat. So you've got some economic arguments you have to win there as well.
Yeah, and there's some interesting companies that trying to counteract that. They're looking at the cost and trying to bring it down. Sorry, we've gone massively off track, but I want to completely change the subject now. I want to talk about work environment. How do you create a great work environment, especially for a small, dynamic, ambitious team as well?
Yeah, I think for a small biotech company setting, and particularly like us, where we really started to grow the company during COVID, and for at least a year and a half, we're hiring people by video, we're running the company by video. It's really important to think very carefully when you hire people, because a small company of 8, 10, 12 people is essentially a small family. So I've been very careful to focus on bringing people in with the right experience, with the right competencies, but also making sure that we have the right cultural fit, so that we don't have anybody who essentially disrupts the cart when the number of people involved in the company is actually that small. I think it's different in much bigger organizations, but I think for a small biotech company, especially if you're going to run it semi-virtually, you're going to meet together as a team infrequently. I mean, we meet together as a team every other week for 3 days. So it's not that we're in the office all the time with each other, and 70% of the time we're interacting by video. So you need to know that everyone can work like that, but also that when we are together, it's actually a great feel, that we've got the right respect, we've got the right listening, and we've empowered everybody so that they know what they're doing, that they're free to do it and get on with it, than need to be referring back. I think that's the way that you get a small biotech company to function effectively. I think if we were much bigger and you had everybody in the office and you had labs, it's quite different. And that would reflect back on past experiences in bigger organizations, which wouldn't work the way that small semi-virtual organizations work. So it's all about people and the dynamics of people, as well as their experience, their background, what they bring in terms of competency.
I couldn't agree more. And I suppose the hard thing is how do you check for those kind of personality fits? For you, is it off gut feeling and just conversations with them, or do you go deeper?
I think you get a good feel in the first 10 minutes when you're talking to somebody, and then you start to delve deeper into not so much their personality, because all of that comes out as you have more conversations. So you start delving into the scientific background or their clinical experience or regulatory experience, etc. And then their personalities show through in what they're talking about, and you begin to form an opinion of, okay, is this person going to fit into the team? Do I believe that to be the case? So yeah, I think it's a little bit of gut feeling and just picking things up as you go. I mean, interview processes take time, so you have multiple rounds of interaction before you finally offer somebody a role, and you've worked it out by then.
Yeah, and that's definitely something you get better at throughout your career. Everyone's made bad hires that just don't gel, and then you learn from there and so on. So yeah, it's just something you improve in, right?
Yeah, COVID taught us actually how to be much better at assessing people down a video screen, because we couldn't meet people. And I hired a couple of key individuals into the company through that period, all based on video interactions, who had actually then turned out to be exactly the people I expected them to be when I met them in person. But the same is true of raising money. I raised money through COVID and had investors that I never met face to face, which is almost unheard of, to actually not be in a room, shake someone's hand, look them in the eye. So I think COVID really taught us how to better use video to evaluate people and understand people than previously, when we weren't so dependent upon Zoom and Teams and other formats.
Yeah, I agree, and it's proven that, you know, if we couldn't do our job from anywhere in the world before, like, you definitely can. And like, from a recruitment standpoint, like that then opens up the amount of people you can then talk to, and the pool of individuals that fit that position. So, you know, you could even afford to be that little bit more picky in terms of the profiles you bring in. So yeah, I agree with that so much. Our final question, which is a question we ask every guest that comes on the podcast, which is, if you could be sat next to anyone at a dinner party, and this person can be dead or alive, who would it be and why?
I mean, there's so many people you could want to sit next to at a dinner party. I guess I'm reading a book at the moment by Jim Watson. So I think maybe he would be a really interesting person to sit next to at a dinner party. I think to talk through that initial sort of solving of the structure of DNA through to everything that's gone on in one lifetime in molecular biology, and the role that he's played there, because he's done an awful lot of things along the way beyond the early stuff that was done around solving that structure. So I think he would be a fascinating person to sit next to. But I think on a more personal note, a person that I would actually love to sit down with is actually my old biology teacher, because I had this fantastic biology teacher at school who got me into science, led me all the way through then to what I've done now. And I haven't seen him since I left school. His name's Jake Cremona. And I'd love to actually sit down next to him, because he'd have lots of stories to tell about other students that came through after me. And I think that'd be fascinating as well. So that's a more personal touch of somebody that I'd love to sit down next to. And I'm not even sure if he's still alive, but you know, that would be great as well.
That's such a cool answer. I love that. You know, someone who kind of shaped what you wanted to go into and all of that. Yeah, amazing. It's been an absolute pleasure to have you on, Andy.
No, it's been a pleasure, Max. Great to speak with you.
Yeah, and everyone, please like and subscribe to the BioHub. And thanks so much again, Andy.
Great. You're welcome. All right.